How do we devise a system to control a concentration of molecules in the blood or extracellular fluids? In this segment, we're going to design a hormonal system to control the amount of anything, in this case iron, Fe, in the blood. The system needs to control two things: absorption in the intestine and excretion in the kidney. Remember that for this sort of system, we can start anywhere. I'm going to start with target cells. I'm going to start with just one target cell in the intestine. When iron is low, we want to move iron that's in the gut into the cell and out into the extracellular fluid. We can do that with a combination of transporters and pumps. We usually want to consider at least one pump. So if we had an active transporter that moves iron into a cell requiring an ATP, then we could have a passive transporter that, as iron accumulates, can be moved out. So we want this to happen if iron levels are low. So our hormone needs to bind, activate a second messenger pathway. So this could be a G protein coupled receptor, similar to one we've got before. We have choices as to how we use this second messenger to change the amount of iron. One way we can do that is by having our second messenger bind to a transcription factor, and the second messenger and transcription factor move into the cell and produce messenger RNA that leads to more protein for the iron transporter and pump. That would be a way to change the amount of iron, to increase the amount of iron in the extracellular fluid. Now we need a way to detect the amount of iron and produce the hormone. So we're going to go back to the beginning. Here's a cell, extracellular fluid, and we'll invent a receptor for iron and result in release of the hormone. The amount of hormone in this case is indicating how much iron we've got. So now let's try and connect these two. So we have a cell, binds iron, we get a second messenger cascade that leads to a series of events that result in hormone release. The hormone gets into the blood, but of course it only affects cells that have receptors for it. And so here's our cell binding to a cell in the intestine. And we want, when there's high iron, we want to have fewer iron transporters, and we want to have fewer iron pumps. So in this case, we would want our second messenger bind to a transcription factor that then moves into the nucleus. And if we have a gene here for an iron pump, and elsewhere for an ion transporter, then we want, with more hormone, we want to decrease movement of iron out of the gut and into the extracellular fluid. So we'd want fewer iron pumps and fewer iron transporters. So we want these to be repressors. So let's look at our system. When there's high iron, we cause release of the hormone. The hormone moves into the blood, moves out anywhere, binds to cells that have the receptor, in this case intestinal cells. The intestinal cells can express iron transporters and iron pumps. When there's high iron, we want to take less iron out of the intestine and less iron into the extracellular fluid. So our second messenger binds to transcription factors that repress transcription, leading to less messenger RNA and less protein. So what about in the kidney? In the kidney, again, we'd have a pump that can pump iron into a cell and a passive transporter that can release it into the urine. In the kidney, when iron is high, and our hormone binds, if iron is too high, we want to get rid of the excess. And so in this case, we want our second messenger We want our second messenger to bind to a TF in the regulatory region for iron transporter and iron pump. And when iron levels are high, we want to be excreting more. So in this case, it's a different transcription factor. We want both of them to be enhancer. So think through our system. When iron is high, we release hormone. When it gets to the kidney, the hormone binds, activates a second messenger. The second messenger binds to a transcription factor, which enhances transcription of messenger RNA for the iron transporter and pump. We get more mRNA and more pumps and transporters. So when iron is high, we successfully get rid of excess iron into the urine. Let's go back and check our original question. We want a hormonal system that controls the amount of iron, controlling absorption in the intestine and excretion in the kidney. When iron is high, it's going to suppress absorption of iron in the intestine, and it's going to increase excretion of iron in the kidney. When iron is low, it's going to fail to repress the absorption of iron in the intestine, and it's going to fail to increase excretion of iron from the kidney. So our system will work correctly. It detects, we have hormone release, we have action on target cells. There's an effect, we change the amount of iron. There's feedback, the change in iron affects detection and therefore hormone release.